All right, well, I'd also like to thank Paige and John for inviting me to talk on our work, which really at its heart focuses on how peroxisomes and mitochondria cooperate in order to combat lipid-induced insulin resistance. So when we think of lipid-induced insulin resistance, there are typically two models that people think of. One model is you get this excess lipid that floods its way into the mitochondria, and that overwhelms its capacity to break it down. And that the overwhelming lipid flux into the mitochondria induces a stress which feeds back to inhibit insulin sensitivity. And that's the mitochondrial lipid overload model. The second model is when you get excess lipid, you get accumulation of acyl-CoAs, diacylglycerides, ceramides, and that these can directly interfere with the insulin signaling cascade, and that's referred to as the lipotoxic model of insulin resistance. And my argument is that regardless of whichever model you prefer, I think that peroxisomes have the capability of alleviating lipid-induced insulin resistance because they can provide an alternate route for lipid disposal that can both minimize the lipid excess that goes into the mitochondria as well as limit the accumulation of these excess lipotoxic lipid species. And this model really is the central focus of our lab. So with that being said, I must admit that I have a little bit of bias that I favor the mitochondrial lipid overload model because I was a postdoctoral fellow in Deb Muyo's lab and contributed to studies that helped develop the model. And the key pieces of information that really led to the development of the model were when we used Etamoxor and malonyl-CoA to inhibit lipid entry into the mitochondria by inhibiting CPT1, which is the rate-limiting step. And so when we reduced lipid uptake into the mitochondria, we found that that improved insulin sensitivity and normalized glucose homeostasis. So naturally, we were pretty excited about this, but I was also thrilled when I got to Pennington because Randy Minot had developed a muscle-specific CPT1 knockout animal, which would allow us to test this model more directly. And just like we would have predicted based on this model, you can see that the muscle-specific CPT1 knockout mice have improved glucose tolerance as well. So, naturally we were excited about this. We wanted to pursue it a little bit further. So one of the first studies that we did was we isolated mitochondria from these animals, and we measured palmitate oxidation in the presence and absence of Etamoxor, which is the CPT1 inhibitor. And as you can see in our control animals, Etamoxor effectively decreased palmitate oxidation, whereas that regulation is lost in the knockout animals. And in the knockout animals, we also see about a 60 to 70% decrease in total palmitate oxidation in the mitochondria, which basically confirms the genotype. So we next wanted to extend this to look at palmitate oxidation in tissue homogenates. And once again, we did the same assays, and you see that Etamoxor inhibits palmitate oxidation in the controls. That regulation is lost in the knockout animals. However, what's interesting about this particular piece of data is the fact that we get a much less robust decrease in palmitate oxidation. In fact, it was only about a 20% decrease in palmitate oxidation. And when we saw this data, Randy's group immediately panicked and thought that they weren't getting enough decrease in oxidation. However, I was pretty excited because it reminded me of my days in graduate school. I know this model is impossible for everybody to read out there. It's simply a reminder of me to tell you what the data showed. So what we showed with those particular studies is that when we inhibited lipid entry into the mitochondria by inhibiting CPT1, that lipid could be trafficked to the peroxisome where it can be broken down, and then the products of peroxisomal oxidation can then enter the mitochondria independent of CPT1. Therefore, when you inhibit CPT1, peroxisomes have the capability of rescuing oxidation. And I thought that that's probably what was happening in our tissue homogenates. So we looked at markers of peroxisomal adaptations in these animals. And as you can see, we saw several genes of peroxisomal function increased. And then we also measured lignoceric acid oxidation. And the reason that this is important is because lignoceric acid is a 24-carbon, very long-chain fat. And mitochondria are incapable of processing lipid species that are 22 carbons and above. In fact, a peroxisome has to do that. So when we measure lignoceric acid oxidation, what we're effectively getting is a very specific and very sensitive measure of peroxisomal function. So what you see here is that there's a significant increase in peroxisomal function in these CPT1 knockout mice. So I'm not going to show a lot more data about those particular animals, but the muscle-specific CPT1 knockouts had a very robust whole body phenotype. It was very interesting, so we decided to run a SAGE analysis on them. And when we were doing the bioinformatics, I found it really interesting that the shifts in gene expression in those animals were associated with decreased risk of a lot of different metabolic diseases, as well as GI diseases, cardiovascular disease, and even premature death. So the CPT1 deficiency in muscle, it really improved the metabolic health of these animals. So we were pretty excited about that, but what really made me excited is when we did the gene set enrichment analysis, we saw that the peroxisomal pathways were the second most robustly upregulated metabolic pathways in these animals. So naturally, that just made me have to ask the question, is the upregulation in peroxisomes that we see in these animals critical for the improvements in metabolic health that we see? So in order to accomplish this goal, we developed a muscle-specific peroxisome-deficient animal model in order to test that. So how do we do that? I contacted Miriam Bias at the University of Leuven in Belgium because she had developed an animal model where the PEX5 gene was floxed. The reason this is important is because while peroxisomes are really important of a lot of different functions, they're still fairly crude in the sense that roughly 80 to 90% of the proteins that enter the peroxisomal matrix are delivered there by a single taxi service, and that taxi service is PEX5. So PEX5 binds to these proteins and brings them into the peroxisomal matrix. Therefore, when we knock it out, what we effectively have is a peroxisomal ghost. It still has the lipid structure, but is virtually devoid of enzymatic activity in the matrix. Now again, we want to develop muscle-specific knockouts, so we bred those animals to, bred them to animals that had the Cre recombinase gene driven by the MLC1F promoter, which again is specific to skeletal muscle but doesn't affect the heart. So that allowed us to get this muscle-specific peroxisome knockout animal. Now again, I also am very curious as to whether the upregulation of peroxisomes that we see in the CPT1 knockout mice is important for the improvements in metabolic health. So we also bred our PEX5 knockout animals to the CPT knockout animals in order to develop the double knockouts. So one of the first things, or one of the most important things that I am interested in is how peroxisomes in muscle affect glucose homeostasis. So we performed a glucose tolerance test, and just like Randy's group, our cohort of CPT1 knockout mice had improved glucose tolerance. Now I guess I should probably back up for a second and say that when we present this data, it's very common for people to sit there and think, look, you're limiting the ability of the muscle to use fat as a fuel, so naturally it's going to compensate by increasing its utilization of other fuels. And I totally agree with that. I think that's probably what is contributing significantly to this improvement in glucose homeostasis. But my next question was, what happens when we knock out the peroxisome? Do we see a similar effect? In fact, we definitely do not see a similar effect. We actually see that the peroxisome deficient mice have impaired glucose tolerance. And this is actually what was predicted by our model. So naturally, the next question is, since we developed the double knockout mice, what happens? What we see when we knock out lipid metabolism through both the mitochondria and the peroxisome is that the curves overlay almost identically. So how do we interpret this data? So we're interpreting it two different ways. Actually, I mean two different things, or two independent things. One, when we knock out peroxisomal function in muscle, that impairs whole body glucose homeostasis. However, that can be rescued if you limit excess lipid metabolism through the mitochondria, because you don't see that in the double knockout mice. Secondly, the improvements that you see in the CPT1 knockout mice in glucose tolerance require the ability to upregulate peroxisomes, because also when you knock out peroxisomes in those animals, you negate the improvements in glucose homeostasis. So this really emphasizes the importance of peroxisomes in helping to maintain whole body glucose homeostasis. So we wanted to delve a little bit deeper into the mechanisms. So one of the first things we did was take tissue homogenates and measured lignoceric acid oxidation to get an idea of what's happening with peroxisomal function. And again, just like we saw in the original cohort, we saw about a 25% increase in peroxisomal oxidation in the CPT1 knockout mice, indicating upregulation of peroxisomes. And as you'd expect, when we knock out PEX5 in our PEX5 knockouts and double knockout animals, we see significant decreases in peroxisomal oxidation. And then this is more confirmatory of the genotypes than anything. But I should mention that, so lignoceric acid, again, is a very long chain fat. Those aren't particularly abundant in our diets or in circulation. So we wanted to see what happens with the more common lipid fuel. So we measured palmitate oxidation again, and almost identical to the original cohort. We saw about a 21% decrease in palmitate oxidation. So, the next step was to see what happens when we knocked out peroxisomes. And, I should mention that I didn't truly know what to predict here, because while peroxisomes are perfectly capable of handling palmitate, they aren't necessarily required because the mitochondria can handle them. So when we got the data back, I can say that I was pleasantly surprised by the fact that we actually saw about a 29 to 30% decrease in palmitate oxidation. And the reason this surprised me was because I wasn't expecting this decrease to be as significant or even more significant than what we saw when we knock out CPT1. So this gives you a pretty good idea that the peroxisomes are pretty important in lipid metabolism in skeletal muscle. So while I was surprised by the extent of this, what shouldn't be a surprise to anybody based on these two graphs is that when we knocked out lipid metabolism through both the mitochondria and the peroxisome, that we get a very severe impairment in palmitate oxidation. So, that gives you a good idea of what's going on with lipid metabolism in these tissues, but we wanted to extend that further and look at metabolism of other substrates. So we measured oxidation of the branched chain amino acid leucine, and what you see in the CPT1 knockout mice is they have about a 22% increase in the capacity to break down leucine. And, again, we think that that's simply because of the limitation in the ability of the muscle to use fat, that they're compensating to increase utilization of other substrates. So, do we see something similar in the peroxisome deficient mice? The answer to that is no. We actually see that those animals have about a 25% decrease in their capacity to utilize leucine. And I think this is really interesting, but I'm going to come back to that point in just a second. So naturally, what happens in our double knockout mice? The question would be, do these effects wash each other out? The answer to that is, once again, no. In fact, what we see is a much more robust increase in the oxidation of leucine as a substrate in the double knockout mice. And again, I think what is really happening is because the impairment in fatty acid oxidation in these double knockouts is so severe that they're upregulating their ability to use any other substrates much more so than the CPT1 knockout animals. And we confirmed that as well, looking at PDH activity and pyruvate oxidation. I'm not showing those graphs because they're almost identical to these. So this data really indicates that when we limit lipid metabolism, lipid uptake into the mitochondria in either the CPT1 knockout or the double knockouts, the system responds by increasing its ability to use both carbohydrate and amino acid-derived fuels. However, what's kind of interesting is when we knock out peroxisomal function, we actually also saw a 25% decrease in oxidation of these substrates. Now remember, we saw about a 30% decrease in palmitate oxidation in the muscle of these peroxisome deficient mice. We're also seeing decreases of about 25 to 30% in their ability to use carbohydrate and amino acid-derived fuels. So to me, that's really telling me that in the absence of peroxisomes, the mitochondria suffer. They're being overwhelmed with too much lipid substrate, and they're starting to decompensate. And we're developing cohorts in order to test that more directly right now, but there is precedent to think that this could be accurate because Miriam's group developed a liver-specific peroxisome knockout animal. And when they looked at the mitochondria, they saw that the ultrastructure, they had swollen mitochondria, the cristae were malformed, they had a severe impairment in their ability to generate a membrane potential, and they had profound reductions in activity of complex 1, complex 3, and complex 5 of the electron transport chain. And I predict that we'll see something similar in the skeletal muscle. So naturally, if we see decreases in mitochondrial content and function, that could mean a bunch of things. It could mean that the absence of peroxisomes is by default flooding the mitochondria with excess lipid. It could mean that the mitochondrial lipid membrane structure is changing. And we're going to be testing all those things, but something that we found that I wanted to bring to everybody's attention came when we looked at the gene expression analysis in these animals. So what this graph shows is basically just confirmation of the genotype. When we measured CPT1 expression, you see that it's lower in the CPT1 and double knockout animals and unchanged in the PEX5 knockout animals. When we looked at PEX5 expression, you see that PEX5 is lower in the PEX5 knockouts and the double knockout animals, but it's actually increased in the CPT1 knockout animals, again, supporting increased peroxisomal compensation in those animals. So the next step that we took was to measure expression of several genes that are involved in fatty acid catabolism in the peroxisome and as well as in the mitochondria, or at least genes that favor fatty acid metabolism. And what we saw here was kind of interesting. So you see that in the CPT1 knockout animals, which are these white bars, there's a significant increase not only in the peroxisomal genes, but also in mitochondrial genes that favor fatty acid metabolism. Whereas we don't see that type of compensation in either the PEX5 or the double knockout animals. So the way that I'm looking at this data suggests that when we knock out peroxisomes, the system just doesn't recognize that as a problem. It's not trying to compensate to increase the oxidation of these lipids. However, when we knock out mitochondrial lipid entry and mitochondrial lipid oxidation by default, the system then sees that as a severe problem. It tries to upregulate anything it can in order to enhance the utilization and disposal of these lipids through either peroxisomal or mitochondrial genes. However, this upregulation in response to CPT1 deficiency absolutely requires the peroxisome because this upregulation is completely negated in our double knockout mice. So it appears that peroxisomal function is required in order to upregulate this transcriptional response in order to induce fat metabolism. And we got a hint of that when we exercise trained these animals. So what we see when we measured lignoceric acid oxidation in our control animals, we saw about a twofold increase in peroxisomal function, whereas that was not seen at all in our knockout animals, which is completely what you'd expect and predict. However, when we looked at palmitate oxidation, this is what I found fairly interesting, is the fact that in our control animals, palmitate oxidation increased about 50%, but the ability of exercise to increase palmitate oxidation capacity in the knockout animals was significantly blunted. So collectively, these last two pieces of data led me to develop this next model. So from our studies, we know that when we knock out CPT1 and limit lipid entry into the mitochondria, data that I didn't show but is published in PNAS, shows that this induces a mitochondrial energy deficit signal. There's activation of the AMP kinase pathways, PGC1 is upregulated, and we all know that activation of those pathways can induce this gene transcription program. However, our data would suggest that peroxisomes absolutely have to be functional in order to observe these increases in fatty acid oxidation gene transcription. So I think what is happening is that when whatever model you end up choosing, when lipid is in excess of what the mitochondria can utilize, it gets diverted to the peroxisome and it undergoes peroxisomal metabolism of some sort, and that the peroxisome may be producing some as yet unidentified lipid metabolite that can serve as a ligand for PPAR responses, which can then potentiate the induction of these fatty acid oxidation genes. Now, I know I may run the risk of being Indiana Jones and chasing after an artifact here, but I think it's exciting, potentially novel, and potentially important. So we are pursuing that through a pilot and feasibility grant that we recently got, as well as we're running non-targeted discovery-based lipidomics profiling in order to see if we can start honing in on a subset of lipid metabolites that may be facilitating this response that are derived from peroxisomes. So while this is risky, the concept isn't necessarily unprecedented because Clay Semenkovich's lab recently showed that in white adipose tissue, that lipid handling, that lipids can go through the peroxisome and they can produce ether phospholipids. And these ether phospholipids can be very potent agonists for PPAR gamma, And in that particular tissue, it activates this adipogenic program. So our model is conceptually similar, but fundamentally different in the sense that where Dr. Semenkovich's model suggests that lipid handling through the peroxisome can drive an adipogenic program, our model would suggest that lipid handling through the peroxisome can drive a lipid breakdown pathway. So, if this model is accurate and if our model is somewhat accurate, it would kind of suggest that peroxisomes sort of lie as a central integration point that can dictate whether the tissue goes into a lipid storage program or lipid breakdown program. And, if so, then, maybe we can manipulate metabolism through the peroxisome in order to sway, one way or another, whether we want more lipid storage or more lipid breakdown. So that's kind of where our lab is heading. But with that being said, the take-home messages from the talk really are, so I think we have pretty compelling evidence to at least suggest that peroxisomes in skeletal muscle do help maintain whole body glucose homeostasis. Because when we knock out peroxisomes in the muscle, that impairs glucose tolerance. However, that can be restored if you limit mitochondrial lipid overload by inhibiting CPT1. Likewise, when in the CPT1 knockout animals, it seems like the improvements in glucose tolerance, at the very least, are pretty dependent on their ability to enhance peroxisomal function. I also found it fairly interesting that when we deleted peroxisomes, that there was about a 30% decrease in palmitate oxidation, which was to at least a similar, if not greater extent than we saw when we knocked out CPT1. And that at least partially suggests that peroxisomes could be very important in overall fat metabolism in the skeletal muscle. However, I think what the major player is the fact that I think peroxisomes are really helping protect mitochondrial function. So when we knock out peroxisomes, they become impaired because we saw not only decreases in fat oxidation, but also decreases in carbohydrate and amino acid oxidation. Also thought it was really interesting that when we knocked out peroxisomes, the animals seemed to be incapable of generating a transcriptional response to activate fatty acid metabolism pathways. And if that ends up being true, it would suggest that peroxisomes may be really important in regulating metabolic flexibility. And finally, I think collectively this information emphasizes the importance of peroxisomal function in muscle and really suggests that if we can design some strategies to regulate peroxisomal function in muscle, that they could offer therapeutic potential. So with that being said, I definitely have to thank everybody. Emily and Roger, who are no longer in the lab, but they did a lot of the work that I showed today. Definitely have to thank Randy Meinetz's lab for allowing me to be involved in the CPT1 knockout studies. Also have great faculty collaborators and core facilities at Pennington. Got my interest in peroxisomal biology in graduate school in Ron Kortwright's lab, and then continued my training in Deb Muyo's lab as a postdoctoral fellow. Also have to thank a tremendous debt of gratitude to Miriam for allowing us to use the PEX-5 floxed animals, as well as the previous and current sources of funding. And with that, I'll be happy to take any questions.